Okay. Hello everyone, my name is Anusha and I am a PhD student at Uppsala University. Now I'm going to speak about the oral microbiome during pregnancy. So let's begin with the introduction to oral microbiota. It's nothing but it consists of microbes inhabiting the oral cavity, which makes it the second most complex ecosystem in the human body. So the oral microbiome varies according to the diet, dental hygiene, lifestyle choices such as smoking, and physiological changes such as pregnancy. However, our focus is on oral microbiome during pregnancy. Well, it's known that a balanced oral microbiome is important for healthy pregnancy. Dysbiosis or imbalance in oral microbial composition may lead to pregnancy-related complications. Well, there are several studies has been done on oral microbiome in pregnant group, but they use 16S rRNA sequencing technology, which doesn't provide functional insights or species-level resolution. We used shotgun metagenomics to compare the salivary microbiome of pregnant and non-pregnant group and to analyze the taxonomic and functional composition. So our study consists of two cohorts, Mimans, which is non-pregnant, and Palmi, which is pregnant. So several samples were removed due to factors such as excessive drinking, smoking, and antibiotic usage. So finally, we had 73 non-pregnant and 42 pregnant samples. Further, the pregnant samples were divided into vaginal delivery and C-section. This was because the fasting time before the sample collection varied between these participants. And non-pregnant samples were categorized into different phases of menstrual cycle. Saliva samples were collected from the participants by giving certain instructions using saliva gene collector. Next generation sequencing was done, and then the taxonomic annotation was performed using Kraken and Bracken tools. Functional annotation was based on KEGG module. In addition to this, we also used qPCR to measure the absolute abundance of selected oral bacteria. Statistical analysis was performed in R, and alpha diversity was measured in terms of richness, evenness, Shannon entropy, and beta diversity was based on adjacent distance. In addition to this, we also performed Permanova and Anacomb-BC. We also used Sokransky color complex on our data. It is nothing but categorizing the periodontal pathogens according to its association with periodontal diseases. So it was classified as shown in this figure. So the blue, green, yellow, and purple group were compatible with oral health, whereas the red and orange group was associated with inflammatory periodontal diseases. So this table displays the demographic characteristics of two cohorts. So the pregnant and non-pregnant cohort were almost similar, but the pregnant samples were more older than non-pregnant, whereas and they were more likely to have previous biological children. And this is the violin plot representing the alpha diversity. So we observed a significant difference in terms of Shannon diversity between pregnant and non-pregnant samples. We also observed in terms of richness also, we observed a significant difference. But in terms of evenness, there was no significant difference. And we performed the same analysis on categorized data of pregnant and non-pregnant group. We couldn't find any significant difference in the non-pregnant group, whereas in the pregnant group, that is C-section and vaginal delivery participants, we could find a significant difference in terms of richness and evenness, but there was no overall diversity. So in short, the pregnant women's saliva had lower diversity and richness than the non-pregnant, but unaltered evenness. So this is the alluvial plot showing the proportion of reeds in each cohort assigned to each phylum and genus. We couldn't find any significant difference at the genus or phylum level. This is the beta diversity plot calculated by adjacent distance. So the beta diversity revealed that there was a significant difference between these two groups, and the variation was due to the lower relative abundance of certain species. Species such as like Leptotrichia species, Neisseria species were like relatively less abundant among pregnant group. We also performed the Sokransky color complex, and interestingly, we found that the red group, which is associated with periodontal pathogens, were more abundant in pregnant group when compared to non-pregnant, whereas the green group, which is associated with oral health, is less abundant in pregnant group. We also performed the qPCR measurement for the selected bacterial pathogens, and we observed that Fusobacterium nucleatum was less abundant in pregnant group, whereas the Porphyromonas gingivalis, which is associated with periodontal disease, was more abundant in pregnant group. So this is the differential abundance analysis, and it's a volcano plot. And here the color is assigned according to the phylum the species belongs to, and we observed that 77 taxa were significantly different between these groups. And here you can see like 21 groups were, 21 taxa in non-pregnant was like twofold higher, whereas Prevetola dentalis and Pseudomonas aeruginosa, these two species were twofold higher in pregnant group. Similarly, we also performed the functional modules. And here the color for the module was assigned according to the Keck pathway it belonged to. And we observed that there was 55 functional modules differed between these two samples. And here you can see two modules. These were threefold higher in non-pregnant samples, and threefold higher in non-pregnant samples, and they were associated with xylene and toluene degradation. So in conclusion, so we found that the reproductive stage affected the salivary microbial composition, and lower oral microbial diversity and richness was observed in pregnant when compared to non-pregnant group. Pro-inflammatory species such as Sokransky red group was more prevalent in red group, in pregnant group, and the xenobiotic degradation, like the xylene and toluene, the potential for the degradation of xylene and toluene was reduced in pregnant group when compared to non-pregnant group. We also observed a bidirectional interaction where the host hormonal signals can influence the oral microbial behavior, whereas the microbial metabolites can impact the host health. So future research should focus on maintaining the oral health and minimize the dysbiosis during pregnancy. So I would like to thank all the collaborators involved in this work, and I would also like to thank Novo Nordisk Foundation for their support on this presentation. Thank you.
Thank you very much, Anusha. We have time for one question. I will ask you a question, though. How much do we know about the physicochemical differences of the saliva between the pregnant women and the non-pregnant ones? So, do you know whether there are differences between the chemical composition of the saliva that could be explaining the differences you observe in the drop of the diversity?
Yes, I mean, the hormonal changes can surely will impact the oral microbial composition or in other way. So we really don't know about that.
Okay.
Yeah.
Good.
Yeah.
Thank you. Yeah. So we will continue with the next speaker. Thanks, Anusha.